Essentially, the beta-2 adrenergic receptors or the beta-adrenergic receptors are the best understood G-protein coupled receptors from both a structural and a functional perspective. I don't have the time to go into detail now, but the understanding the molecular structure of any protein can be resolved via crystallization, basically forming a crystal of the protein, but this is very difficult to do with membrane proteins. Well, the fact is that the membrane proteins have been crystallized, and particularly the beta-adrenergic receptors have been crystallized and studied, and not only that, through a complicated set of methods and techniques, they've been able to study even the ligand-bound structure of the G-protein, the beta-adrenergic receptor, in a relatively recent paper that is actually shown on this slide. But this is not the main thing. Essentially, I want to relate on what is the effect of the beta-adrenergic receptors. Beta-adrenergic receptors, again, best studied G-protein coupled receptors, and they can serve us as a sort of an example for this lecture. Three subtypes of beta-adrenergic receptors: the beta-1, the beta-2, and the beta-3. And what you see here is that their primary G protein is the GS, the stimulatory protein. That means that they will result in an increase in cyclic AMP. But beta-2 and beta-3 adrenergic receptors are known for being promiscuous. That means that they can both activate the GS but also the GI pathways, and that will depend on the expression, of course, of the different G protein subtypes. You see, as the tissue distribution, beta-1 and beta-2 are primordial in the heart in terms of stimulating heart contractility. The beta-3 adrenergic receptors are mostly located to the adipose tissue, in what's called the brown adipose tissue that produces heat. The primary effectors in all three types is the adenylate cyclase, and then leading to an increase of cyclic AMP if it's a GS pathway, or a decrease if it's a GI pathway. What you notice here is that besides the cyclic AMP, it also lists protein kinase A, so it's the activation of protein kinase A that is involved. In the human heart, essentially 75 to 80% of the beta-adrenergic receptors are of the beta-1 subtype. There are also alpha-adrenergic receptors in a ratio of 1 to 10 to the beta-adrenergic receptors in the heart, but I'm not going to be talking about them much more. Of course, the beta-adrenergic receptors have a classical role in potentiating cellular processes, in this case EC coupling in cardiomyocytes, contraction and relaxation of the heart, increasing the heart rate, and this is done by the generated second messengers, mostly cyclic AMP. Adrenergic receptors are also involved, however, in the control of cardiomyocyte growth and apoptosis. Here you have a basic sketch of different types of adrenergic receptor signaling. I'm going to focus on the top ones, basically signaling through GS or GI, both of them involved in the production of cyclic AMP, which in turn can activate protein kinase A to induce a cellular response. As you see here, basically it's mostly the beta-adrenergic receptors are involved with the activation through GS of cyclic AMP. As I said in the previous slide, there is evidence that the beta-2 and 3 adrenergic receptors can also be involved with the GI, which is known for alpha-2 adrenergic receptors. In the bottom part, you have alpha-1 adrenergic receptors that actually signal through the GQ pathway. The important thing in this lecture, I want to try to basically combine the role of the beta-adrenergic receptors with the concept of heart failure and how beta-adrenergic receptors are involved in this pathology. Before we go into it, is basically that heart failure is defined as an inotropic failure. Inotropic referring to the contractile capability of the heart. So if there is inotropic failure, basically the result is it's an inability, a decreasing pumping efficiency. And this could actually be caused by many different situations. One of them is hypertension. but also the injury of the heart muscle. For example, a heart muscle that has undergone an infarct, that means that a region of the heart muscle have died, is not going to be able to produce the same kind of force. When a heart starts failing, as a result of this failing, there is actually changes in muscle structure as well. Changes in muscle structure as well that result in a heart that is getting weaker and weaker with time and resulting in a worse. And what you can see in these two pictures is a heart failing heart in your left hand side versus a more normal heart in your right hand side. And what you can appreciate in this picture is essentially the thickness of the left ventricular wall. This thickness in anatropic failure after remodeling is thinned out. And notice also that the size of the chamber is much increased in heart failure. This is a heart that is not being able to pump out, it accumulates blood, the heart muscle is failing, it's actually thinning, there is hypertrophic growth of the heart is enlarged but is not pumping. Unlike what you have in this situation, an efficient heart that is able to pump, thicker walls, and effective in actually emptying. Well, if we think in terms of what's happening with heart failure, what is leading to heart failure, this has to be seen as a two-step process. The heart, in regular conditions, is going to respond to injuries, for example, infarct, but also other challenges that are actually, these injuries result in a decrease in cardiac output. When that happens, the heart wants to return cardiac output to its value levels. And how does it achieve that? Via stimulation of the sympathetic nervous system, which will produce noradrenaline. Noradrenaline is actually targeting the beta-adrenergic receptors that, as we have seen, stimulate GS, produce or induce AC, produces cyclic AMP, and that will in turn result in an increase in the force and frequency of the heart with an acute improvement of the original situation. So through beta-adrenergic stimulation, the heart is trying to compensate for different situations that can be acute or chronic. The problem comes when essentially this loop of improvement basically is not accompanied by an improvement because the heart muscle is actually damaged. When this cycle is stimulated over and over and over and over again, what happens with the alpha-adrenergic receptors? Presumably, the alpha or the beta-adrenergic receptors are down-regulated, are desensitized. The result here is that this internal loop that was an acute compensation ends up running into the external loop, which is the chronic situation. In the chronic situation, the heart starts consuming more, there is apoptosis, there is hypertrophy, there can be arrhythmia, and then this is a vicious cycle. The further and further sympathetic stimulation no longer is able to trigger the acute improvement. So in the long term, this heart, because it's chronically stimulated, because the beta-adrenergic receptors are chronically stimulated, there is a worsening in cardiac function as a result of the chronic desensitization of the beta-adrenergic receptors. Beta-adrenergic receptors are by no means the only culprit in heart failure. I'm not trying to say this. There are many other reasons why the heart can be failing, but this is an important one to take into consideration. So essentially, heart failure associated with chronic increase in circulating catecholamines. Remember, this is an adaptive response. The heart tries to compensate, but in a chronic scenario, it cannot do that. Then it leads to beta-adrenergic desensitization and downregulation. We know, as a matter of fact, that beta-1 adrenergic receptors are downregulated and are uncoupled in heart failure. In a failing heart, there are fewer adrenergic receptors that are actually working. We also know that GRK2 is overexpressed in heart failure. Remember what was GRK2? GRK2 is involved in homologous desensitization. It's a kinase that will basically phosphorylate the beta-adrenergic receptor and trigger a smaller response from it. The interesting thing, though, is that if we consider what can we do with it, and this has been a long-term problem that we have known, It's well known nowadays that treating the heart, treating the body with inotropic drugs, drugs that will stimulate the heart to pump more, will not improve the prognosis of heart failure. Essentially, when inotropic drugs are given, what we would be doing is keeping the circle, the vicious circle I outlined earlier, going. So inotropes do not work. Well, then the question becomes is what can we do? And one option that it was believed or it was tested a few years ago, although counterintuitive, was to test or to treat patients with heart failure, patients with left ventricular dysfunction with beta blockers. What is a beta blocker? A beta blocker is an antagonist of the beta-adrenergic receptors. That means it will antagonize the receptor and decrease the response. Do you think that this is something that would be advisable to do? What would be the expected result of this? If beta blockers, as I said, antagonize beta-adrenergic activation, prevent the positive inotropic effect, prevent the positive chronotropic effect, and prevent the activation of the sympathetic nerves in the heart. Would it be advisable? Beta blockers, of course, depress cardiac function. Beta blockers have been for a long time used for controlling high blood pressure. So it works in decreasing high blood pressure. Would they work against heart failure? Of course, the classical answer should be no. They should not work because they are going to worsen the condition of the heart failure patient. Well, interestingly, Swedish doctor Fredrik Wangstein in 1975 used beta blockers in patients with congestive heart failure. Wangstein basically had to use or decided to use that because he had a group of patients that basically were in terminal phase of heart failure. There was nothing that could be done for them, and they decided to treat these failing hearts with beta-adrenergic blockades. This was published in the British Heart Journal in 1975, actually showing interesting effects. Obviously, and this was expected, patients with heart failure that were treated with beta blockers initially experienced adverse short-term effects. That means that in the short term, these patients suffered from a decreased depression in cardiac function, as would be expected. The interesting thing is that it was shown that the long-term effects were beneficial instead of being bad for the patients. Recent studies have demonstrated that there is a remarkable improvement and there is a remarkable increased survivability of patients with moderate and severe heart failure that are treated with beta blockers. Why? Well, the mechanisms even today are not understood, but it's clear that beta blockers are working or are advisable. And in 1990, basically about a bit under 20 years after the first publication by Wangstein in 1975, it became mainstream therapeutical practice to start administering beta blockers to heart failure patients. The interesting thing in this respect is basically to acknowledge that the heart failure, one of the components of heart failure, is actually the decreased sensitization of the heart to beta-adrenergic stimulation. It's relevant then to understand or to study these mechanisms. Can we do something else? Well, this is something that I was involved a few years ago with my first PhD student. This first study was published in 2009. Is that basically we learned that perinatally, beta-adrenergic receptors from embryonic hearts did not desensitize despite elevated catecholamine levels. So it seems that the embryonic heart, in this case we were working with chickens, but it has been reported for rats and mice as well, that embryonic hearts or the beta-adrenergic receptors in embryonic hearts we do not desensitize. We thought that this was interesting, and actually we carried out work on that. Isa Lindgren defended her PhD thesis without being able to fully answer why is it that beta-adrenergic receptors do not desensitize? And that's something that we are still interested, or I'm still interested in working, but we're just not having any financing for it at the moment. Basically, what ESA was able to demonstrate, as this data shows, this is just a reminder that this was data collected on chickens, is that when we looked at the sensitivity of the heart muscle to actually adrenaline, and this is shown as the PEC50, the sensitivity to adrenaline, it means that the concentration of adrenaline required to stimulate the heart muscle to a given extent. This is what we saw. Notice the open and the closed bars. The open bars were animals incubated normally, and this is done after 2 weeks in incubation. This is an embryonic heart. After 19, this is also an embryonic heart. After 2 weeks and after 5 weeks. So these are in chickens, in actual chickens after hatching. Notice that what we have as the open bars, these are animals incubated under hypoxic conditions. This means animals whose hearts were incubated in less oxygenated conditions. What happens in this scenario? Notice that these hearts, the chronically hypoxic hearts, are hearts that are stressed during the entire incubation period. These are hearts that during hypoxia they release higher levels of catecholamines. They would be chronically exposed to catecholamines. Instead, we do not see desensitization. Why don't we see desensitization? Because the concentration of noradrenaline needed for a given response is less. This is a -8, -7, -8 molar. So as you go higher, it's less concentration of noradrenaline. So in the embryonic heart, basically you need less catecholamines, less noradrenaline to actually cause the same response. What do you see in post-embryonic hearts? The opposite effect. So this is basically evidence of what we were saying, that embryonic beta-adrenergic receptors do not desensitize, but post-natal adrenal receptors desensitize. Well, this is one point for further discussion, because I'm not going to mention more of this. Sorry guys, I just had to move my recording studio. There were some children coming to the swimming pool, and I thought that it would start splashing my computer, so I had to move away. Anyway, what I was saying is that receptor signaling is actually far from simple. So far I've given you a picture in which basically the activation or inactivation of beta-adrenergic receptors leads to different types of intracellular processes. I was saying, for example, that overstimulation with adrenaline or noradrenaline may lead to desensitization of the receptors. Well, that's fair enough, but there is much more new data that we have nowadays that is actually giving us an idea of the complexity. And this paper by Nikolaev and others from 2010 is particularly giving in that respect. Let's see if I can get the pointer running. Pointer options. Where is pointer? Here you go. In A, or in the top and in the bottom side, you have basically the differences at 3 level, or in different aspects of receptor signaling. What you see in the top part is basically what happens with healthy cells. The picture in the left side is actually taken with atomic microscopy, in which you see the structure of the cell surface, and you see a regularity pattern which is associated with the presence of T-tubules. I'm not sure now if I have to review in a little bit of cardiac cell anatomy, but in cardiac cells you have these T-tubules that are invaginations of the plasma membrane in order to facilitate the transport or the calcium from the extracellular space reaching the intracellular space for contractility. What you see here is that the organization of T-tubules in healthy cells is particularly well structured. You have these deep invaginations and then what is called the crests. What that means is essentially that these cells, there are particular areas, and the question remains is intracellular signaling homogeneous or heterogeneous. In other words, if a cell is stimulated, is the cyclic AMP that is found in this cell changing everywhere in the cell, inside the cell, or just in particular spaces? And what the Nikolaev et al. Data, or this paper is showing, is that there is a particular order into cyclic AMP production in these cells. And this is what you see in this site. What essentially these authors were able to do is track using a specially constructed probe that is sensitive to cyclic AMP. They were able to track cyclic AMP concentrations in different areas or different regions of the cell. And these regions are shown with color, and the color corresponds with the line here. What do you see when a cell is activated, a healthy cell is activated? Notice the black trace. The black trace, this one corresponds with the black trace. Is there a change in cyclic AMP? Essentially, no. Now, take the blue one. The blue one is showing you a clear change in cyclic AMP concentration over time. Actually, this is a decrease in fluorescence, or in the ratio of fluorescence, that indicates an increase in cyclic AMP. So what this is showing you is that in healthy cells, you have a clear compartmentalization of cyclic AMP. Cyclic AMP is actually stimulated in some places of the cell and not in others. Now, if we look at what's happening in a failing cell, notice the difference. In a failing cell, the organization of T-tubules and crests has changed. There is not the same level of organization, and that actually results in a much more homogeneous cyclic AMP productivity. When the cell is stimulated in one spot, cyclic AMP concentrations actually increase throughout the cell. And this is characteristic of a failing cell. Well, what that means is that we can no longer look at receptor signaling by taking a sort of an average approach over the entire cell. We need to look at different domains within a cell in order to understand that better. But this is going to be probably the purpose of a further advanced lecture on adrenergic signaling, because this one here is just taking an end. So thank you very much for listening. This is the end. This is part 2 of this lecture, and in a few minutes we can start with the last one, part 3. Thank you very much for listening.